Okay, so we're learning about cell replication in this section. So we're on to chapter 2, and the study design link is all about binary fission in prokaryotes and the cell cycle in eukaryotes. So those two groups of organisms, their cells reproduce in different ways, and we're going to compare those two different things. So your learning intention for this section is that all cells go through a cell cycle, which differs between prokaryotes and eukaryotes. So your success criteria, you should be able to describe what binary fission is, that's how prokaryotes reproduce, and you should be able to describe the cell cycle, which is how eukaryotic cells reproduce. So this whole section about cells is basically about us answering these kinds of questions. So how do little elephants grow up and develop to become big elephants? And why do some animals shed their skin, like these reptiles here? And it's all because cells need to grow and divide and die off in their cell cycle. So the big idea for this section is that all cells reproduce using asexual reproduction. And asexual means we're essentially cloning, well, the cell is essentially cloning itself. It's making a copy, and it has identical DNA to the parent cell that it started with. And the classic view of asexual reproduction is you start off with a parent cell and it divides and splits in half, and then you've got two new daughter cells at the end. And that's called asexual reproduction because there was no sexual reproduction involved, and the daughters are exactly the same as the parent. So why do cells reproduce this way? Well, there's three different reasons. Firstly, it's for growth, and this happens in multicellular organisms in particular. So humans, plants, other animals. Because if we want to grow, we can't just grow our cells larger. Because remember, there's a limit to how big cells can grow. They have to keep that high surface area to volume ratio so they can survive. So we can't just grow our cells bigger, we're going to have to divide and make more cells if we want to grow from a child to an adult. And it's the same thing with repairing and replacing. If we have any damaged tissue, or we need to replace anything that gets lost, then our cells divide and make new cells to make those repairs. The other option is procreation, and this happens in prokaryotes and other single-celled organisms, and other unicellular organisms. Because for them, they can't make more cells as part of their body because their entire body is just one cell. So if they divide, they've basically become two new organisms. So they've procreated. Cancer is another thing to be aware of. So like this person's skin cancer here, they've got a dangerous looking melanoma there under their eye. And all cancers is just abnormal growth of cells. So the cells, the body can't control them anymore. Normally we control them really tightly, but a cancer and a tumor is when those cells grow out of control and it becomes a really serious issue. So there are heaps of different organisms that use asexual reproduction, not just bacteria and single-celled organisms. And we'll learn most of these in term 3 when we learn more about it. But lots of these species, like this worm here and this hydra, you can cut them in half and they'll regrow into two new organisms. Or the hydra here, these little baby little hydras here, they bud off the side of the parent hydra, and they're all asexual copies, they're clones of the parent. Some lizards can actually do it as well, where they become pregnant without having to reproduce. And plants can do it quite regularly, but of course we're mainly focusing on prokaryotes for this section, which are bacteria. So if you can't remember prokaryotes and eukaryotes, here's a quick review. The eukaryotes, if we're thinking of the 5 kingdoms, the eukaryotes is everything except for the bacteria. So it's the animals, the plants, the fungi, and the protists. And remember, we're the ones with more complicated cells. We have a nucleus in our cells, and we have membrane-bound organelles like mitochondria and chloroplasts in plants. The prokaryotes is only the Monera, which is the bacteria. They have much more simple cells. They don't have a nucleus, and they don't have membrane-bound organelles. Okay, so how do prokaryotes reproduce? Well, they do it by this process called binary fission. Binary just means two, like a bicycle with two wheels, and fission is splitting apart. So it just means splitting into two. It's a pretty simple process. It's pretty fast as well. This is the fastest way that cells can reproduce. It's faster than eukaryotes. And you can see the four-step process down here. So firstly, the DNA, which is in the chromosome, replicates. So that's happened here. You can see there was one chromosome and now there's two. Prokaryotes have a circular chromosome, which is unusual. All the eukaryotes, we have linear chromosomes. So once they've replicated, then the cell gets longer, it elongates, and then those two chromosomes move to each end of the cell, ready for it to be split in half. Number 3 is the septum forms, which is a brand new cell wall. So it's almost like the cell is pinching together and a brand new cell wall forms, ready to cut that cell in half. And then obviously the last step is the separation and that cutting in half. So the septum forms and then it breaks apart, and now we've got two brand new bacteria with brand new cell walls, the identical DNA in their chromosomes, so they're clones of each other. So if we follow and make a little acronym out of that, we've got R, E, S, and S. So RES is the 4 steps of binary fission. The maths of binary fission is really interesting too, because it's doubling every time, so it's exponential growth. So if you haven't learned it in maths, we're just doubling every time for exponential growth. So if you start with one cell, it divides into 2, and then each of those divide into 2, so you have 4, then you have 8, then 16, 32, and so on. So it's 2 to the power of n tells us how many cells there are once they've divided. And that means most bacteria can reproduce with binary fission every 20 minutes or so. So if you leave some bacteria for 8 hours, you can get 16 million bacteria or more just through this binary fission of them dividing and dividing. And that's what someone's done here. They've got a swab of some bacteria and they've just got millions of cells there from a few that they started with. Eukaryotic cells are slightly different. They have a whole cell cycle, which is a bit more complicated than just binary fission. Now this is the overview. The cell cycle is just the events that happen in the life of a eukaryotic cell. It's like the diary that's happening to them every week, or like our timetable at school as we follow our week. So a eukaryotic cell starts off in its G1 stage, G for growth. Then it has the S stage, and S is the synthesis. G2 is more growth. Then we have the M, which is mitosis, you might remember from last year. And then it doesn't show it here, but we have cytokinesis is the last stage. So the G1, the S, and the G2, they're all called interphase, and that's where the cell spends most of its time. So if it's a liver cell, it's just sitting there being a liver cell, making enzymes and detoxifying substances like a normal liver cell does in its day-to-day job. But when a cell needs to divide, then it continues along through the M and the cytokinesis, and then the cell divides asexually through mitosis. So this is an overview of the key of the 3 main stages. The interphase, the main job of the interphase is to replicate and copy that DNA, because we need 2 copies of our chromosomes if we're going to have 2 new cells. The mitosis is all about organizing those chromosomes and separating them neatly into two brand new nuclei. Nuclei is the plural of nucleus. And the cytokinesis part is the chopping the cell in two so that we have a brand new two daughter cells with brand new membranes. Here are a few extra details of the cell cycle. So when we start at the G1 stage, G for growth, remember, this is where the cell is just growing, and it does that by increasing the amount of cytosol liquid in there. And it carries out its normal functions, like I said before. This is also when the larger organelles, like the mitochondria and the chloroplasts and the endoplasmic reticulum, these all reproduce and divide in this stage, because it's going to need double the amount of mitochondria when the cell splits in half. The S phase, this is when the cell copies its DNA, synthesizes it. So chromosomes normally look like a single strand of DNA, and we often draw them like this. After the S phase, this is when they look like that typical X shape that we often see chromosomes as. And so after the S phase, it's got the old DNA and the new DNA that it's copied, ready to be split in half. And we'll learn more about that when we do mitosis. The G2 is growing again, and it also prepares a little bit more for cell division. And then the M is the interesting part, where all the chromosomes split apart and we make a new nucleus. That's our next section. And then the cytokinesis is splitting that cell in half to make two new daughter cells. Different types of cells take different amounts of time to do their cell cycle. It depends on which organ they're in and what tissue they're in, for how long it takes. So your average skin cell takes about 2 weeks. So that means we're shedding all of our skin every 2 weeks and replacing it with brand new skin cells. Our red blood cells we're replacing every 4 months, although it doesn't happen all at once, obviously, it's cell by cell. Liver cells last for quite a while, so 300, about a year to year and a half. Our intestinal lining only takes 4 to 5 days. We're constantly shedding and dividing and making brand new intestine cells. And our muscle cells barely ever reproduce and divide. 16 years before a muscle cell divides and goes through its whole cell cycle. So that's how prokaryotes and eukaryotes cells divide and have their life cycle. And in the next section, we're going to learn how mitosis works in a lot more detail.